Back in March 2020, I did a video on mechanism of action of mRNA vaccines. I got a very good response on that video, but some of you people were quite confused. Two of the most commonly asked questions from you people were, number one, can this vaccine alter your DNA? And the other question was that since this vaccine involves the cytotoxic T cell mediated killing of the muscle cell, is that so that it can cause autoimmune reaction? So in this video, I will try to answer these two questions and I will try to clear all other misconceptions in your mind related with this mRNA vaccine. And I will do so by using very simple analogy so that each and every one of you can easily understand that. Hello, I'm Dr. Aizaz from medicovisual.com. Welcome to this visual lecture. So let's start from a very basic concept that let's suppose this is a cell and it can be any cell of your body. The most important macromolecule of your body, structurally as well as functionally, the most important component is protein. Now the recipe to synthesize protein, the recipe to synthesize each and every protein of your body lies in a special recipe book present inside each and every cell of your body. Now, unlike the traditional recipe books, the language of this recipe book is not English or some other language, but it is a language of sequence of nucleotides: adenine, guanine, cytosine and thymine. So I will not go into details of the nucleotides, but that is what it is. Now this is very important book. And this recipe book, it must be protected even within the cell. So it is not just lying within the cytoplasm of your cell, but it is lying within a special double membrane lined structure. So sort of a locker or safe inside your cell is present that harbors this recipe book. Now the factories to create proteins, they lie outside this locker, safe or vault or whatever you like to call it. Now we cannot move this book outside to this factory so that we can create proteins. That would be dangerous. That may damage this book. So because this book cannot be moved outside of this vault, what happens that there are some special workers who sort of copies one or few recipes on a special paper, and now this paper has to be moved, it has to be transported from here to the factory. Although there are small pores or gaps within this vault, this fairly large sort of paper, it cannot come outside of this vault without any external help. But here comes some special proteins or molecules that transport this paper to outside of this vault. Now, what are these things actually? This vault is actually the nucleus, and this action of transporting this component from the nucleus to outside and vice versa, it is called nuclear transport. Now this recipe book, it is actually your DNA, and this protein that transported the substance, this paper outside, it is the exportin. It is called exportin, and similar to that, when we want to import something inside the nucleus, we need importin. So this is nucleus, this is DNA, this is exportin, and what is this substance? What is this paper? This is actually mRNA. So mRNA is not an alien molecule, it is something that is regularly being produced inside your bodies. And by the way, what mRNA means? mRNA simply means messenger RNA. It is sort of a message that was copied from this recipe book. It was the message that was transcribed from the DNA. Right? So The important thing to understand is that number one, mRNA is very unstable molecule. Secondly, mRNA, once it comes out and it came out with the help of exportin, it cannot go inside, it cannot go back inside into the nucleus. So nucleus is well protected by this double membrane, and the transport of substances across this double membrane is well controlled, and that is what we call nuclear transport. So the point to understand here is that because mRNA cannot just by the way go inside, right? It means that it cannot affect the DNA in any way. So the answer to first question is that as the mRNA cannot touch the DNA in any way, it can never ever alter your DNA. Even if it goes inside the nucleus, it will not in any way change your DNA. So that is your misconception. Please clear your minds that it cannot change your DNA. These factories to synthesize the protein, these are basically rough endoplasmic reticulum. They are called rough endoplasmic reticulum. They have that rough surface because of presence of ribosomes on their surface. Of course, there are free ribosomes as well that have their role in protein synthesis, but for now just ignore that. So what happens that this messenger RNA, it will then go to this rough endoplasmic reticulum, and here it will create the proteins. It will be translated into proteins. After creating few protein molecules, it will be ultimately denatured or destroyed. It cannot stay here forever. It will not continue to make lots and lots of protein. It will be ultimately destroyed. It is very unstable molecule. That is why you need ultra low temperature to transport and store mRNA vaccines. Now what will happen to these proteins? A part of this rough endoplasmic reticulum will bud off and it will go to Golgi apparatus, and then Golgi apparatus will pack these proteins and it will throw them outside the cell through the process of exocytosis. All these proteins, it may be used within the cell as well. Secondly, what happens that whatever proteins that are being synthesized within the cell, they will be processed by proteasomes, and the fragments of these proteins, they will be displayed onto MHC1 molecule. This is sort of quality control system of our body. So whatever thing a cell is synthesizing, it will display on its surface, and when cytotoxic T cell will come, they will look that whether the protein they are synthesizing, it is a normal body protein or some dangerous pathogenic protein. So because it is a normal protein, the cytotoxic T cell will not destroy the cell. It will be very happy and it will go away. So this is a part of immune system. That is what happens normally, right? Now let's see what happens when virus infects our body. So let's suppose this is the coronavirus with its spike protein and nucleocapsid, and within the nucleocapsid will be its RNA, and this cell with which it has attached through the ACE2 receptor, this is, let's say, type 2 alveolar pneumocyte. Now what will happen as it attaches its spike protein with ACE2 receptor of the cell, the nucleocapsid will be dropped inside and the nucleocapsid core will be removed, and the structure that will be revealed is the RNA. So now this cell contains viral RNA. But remember one thing, our body does not have capability to copy RNA. I mean, our body cannot make a copy of RNA from RNA, right? Our body can make RNA from DNA, but we cannot make RNA from RNA. So for that reason, this RNA of coronavirus has the recipe of its own RNA replicase enzyme, right? So this RNA, it will use the host machinery, host protein synthesizing machinery, I mean host endoplasmic reticulum or ribosomes, to to produce its own viral replicase that can make a copy of RNA from RNA. Now what this viral replicase will do is that it will use the viral RNA, it will look upon the viral RNA and it will make lots of copies of this RNA. So let's say one copy created, and then it will move again on this and it will create lots of different copies of this RNA. Actually, the replicative cycle of coronavirus is not that simple. I am trying to simplify it as much as possible. If you want to see the details, you can watch my lecture on replicative cycle of coronavirus. I have already discussed the details, but here I am trying to simplify it as much as possible. So what it did is that it created lots of copies of RNA of coronavirus. Along with that, these RNA, they also contain recipe to create viral proteins as well. For example, they contain recipe to produce viral nucleocapsid protein. I mean coronavirus nucleocapsid protein. So nucleocapsid will be created, and remember, by using host machinery, I mean by using humans machinery. So nucleocapsid will be formed and it will associate itself with one of the viral RNA or viral genome. And one of the mRNA, it will go to the rough endoplasmic reticulum and it will synthesize the viral proteins, the spike protein and other proteins. And this nucleocapsid will go into the rough endoplasmic reticulum, and here this part will again bud off, it will go to Golgi apparatus, and in this way it will pack up like a important protein. Golgi apparatus will pack up this coronavirus particles and it will throw them outside, right? And also it will process and break the viral proteins, and it will present the fragments of viral proteins on MHC1 receptor. Now this time when cytotoxic T cell will come and bind with this MHC1 receptor that is harboring the fragment of viral protein, it will be annoyed and it will throw bombs on this cell. It will destroy this cell. Then what will happen? That these coronavirus particles, they will infect another healthy pneumocyte. So as this coronavirus particle infected this pneumocyte like the previous one, it will also start creating coronavirus particles, and again the cytotoxic T cell, they will be annoyed and they will kill this pneumocyte as well, and in this way there will be lots of pneumocytes that will be affected and many of them will die, and that will lead to loss of lung function. That is called acute lung injury, and that may lead to pneumonia and so on, right? So that is very different from what is happening with mRNA vaccine. Now let me tell you what happens when mRNA vaccine is injected into a normal person who has never encountered SARS-CoV-2 in his or her life. So let's suppose here is the person into whom this mRNA vaccine is injected. It could be Moderna's mRNA vaccine or Pfizer's mRNA vaccine or any mRNA vaccine for that matter. So let's enlarge this area and let's see what happens here. So this vaccine, it consists of basically an mRNA, and outside that mRNA is a lipid nanoparticle. Why lipid nanoparticle? As I've already told you in that video, because if naked mRNA is dropped into the intercellular space, it will be destroyed by RNA. Secondly, this mRNA, it cannot enter into the cell. This lipid nanoparticle is, as the name indicates, it is a very small particle, right? And along with that, it is lipophilic. It consists of different types of fats. So it can pass through the lipid bilayer of your cell membrane and it can enter the cell and it can safely drop this mRNA inside the cell. So here is that mRNA. Now let's see the difference between this vaccine's mRNA and RNA of coronavirus. So this vaccine's RNA or vaccine's mRNA, it does not contain the recipe or gene to synthesize replicase enzyme. So it is not self-replicating. Few particles of this mRNA will sort of infect some of the cells, and that is end of story. It cannot again replicate to create more particles. That is the biggest difference. Remember, it is not self-replicating vaccine. And remember, although there is that DNA present inside your nucleus here, this mRNA, it cannot enter into nucleus and it cannot touch this nucleus in any way. This DNA is like a hard disk containing your important data, but this mRNA vaccine is just like we have inserted a USB drive into your computer, and that's what it is doing here. It is sort of a USB drive that is priming your immune system to learn to fight against COVID-19. And secondly, it does not contain recipes to synthesize any other viral protein. It contains the gene to synthesize only one single protein of the coronavirus SARS-CoV-2, and that protein is spike protein of SARS-CoV-2. Now again, what will happen? It will use the rough endoplasmic reticulum's machinery to synthesize the spike protein, and then these spike proteins, they will be packed up by Golgi apparatus and they will be exocytosed. They will be thrown out of the cell, and along with that some fragments of this spike protein, they will be displayed onto MHC1 receptors. Now this time cytotoxic T cell or CD8 positive lymphocyte, again it will come across this cell, and because it is displaying the fragments of a foreign protein, not body's own protein, so again the cytotoxic T cell will be annoyed and it will destroy this cell. And of course it will destroy the cell under the orders of helper T lymphocytes. I am not discussing the helper T lymphocyte to make the lecture very simple and easy for you to understand, but remember that helper T lymphocyte has a very important role here. So it will throw the granzymes or perforins and it will destroy this cell. And what about these spike proteins that are created? They may flow through blood and go to a healthy pneumocyte, but can they infect a healthy pneumocyte? Unlike the coronavirus particles that may inject their genome into a healthy pneumocyte and infect that pneumocyte, the spike protein, even if it may get attached with the ACE2 receptor of type 2 alveolar pneumocyte or healthy pneumocyte, it can never ever infect that pneumocyte, so it will simply fall off, right? But these spike proteins, it is sufficient to stimulate the B lymphocyte, and this B lymphocyte will convert into plasma cell and it will start secreting lots of antibodies against this spike protein. So that is what we want. We will get a lots of antibodies against spike protein and we will get lots of clones of these cells. That's what we call memory cells. We will get memory cytotoxic T cells, we will get memory B lymphocytes, and also we will get memory helper T lymphocytes that are not shown in this diagram. Now here some people might be worried of that lost muscle cell. You may be thinking that some of these muscle cells are lost. So as some of these muscle cells are lost, does it mean that there will be a hole in your shoulder permanently? Of course not. We have 60 trillion cells in our body and daily lots of cells are lost and then new cells replace those cells. So within a day or two, the cells that were lost during this process, they will be healed and new cells will be formed. That's the end of story. That is not something to worry about. Yeah, for a day or two there will be swelling and redness and pain at the injection site, and that is the known side effect of this vaccine. But of course within a day or two it will heal itself. The muscle will heal itself. So you do not need to worry that it will lead to autoimmune reaction. Of course my friends, this is not autoimmune reaction. Autoimmune reaction does not work like that. So then how autoimmune reaction can occur? Well, theoretically speaking, that let's assume that this spike protein, it closely resembles some of the important body proteins. Let's say some protein within your nervous system. Just for the sake of argument, of course this is not true. For the sake of argument, that let's say this spike protein, it resembles some of your important body protein. So antibodies against this spike protein, they may react against that important body protein and they may start destroying that body protein. That is what autoimmunity is. And this process is called molecular mimicry. But of course, that does not happen with this vaccine. Why I am saying so? The reason is simple. This vaccine has been tested in more than 70,000 trial participants and none of them experienced the autoimmune reaction. So of course, it does not cause autoimmune reaction. So the answer to both of your question is no. It cannot alter your DNA, and secondly, it does not cause autoimmune reaction. Finally, how does this vaccine protect? So let's suppose this person has been vaccinated and let's suppose that he or she has been exposed to coronavirus, and of course he or she is having the anti-spike protein antibodies. So as this coronavirus particle tries to attach with its ACE2 receptor, the anti-spike protein antibody will bind to the spike protein and they will destroy this virus particle, and they will not allow this virus particle to attach with its ACE2 receptor. Secondly, let's suppose somehow it got successful in infecting this cell. As soon as it will start creating the vaccine particle by hijacking the rough endoplasmic reticulum of this cell, cytotoxic T cell will come and quickly destroy the cell, and that is the end of story. Right? So that's how this vaccine works. And don't worry about this lost cell. If few cells are lost, they will be replaced within a day or two. The real problem occurs when lots and lots of cells are destroyed and even then virus is not contained. And that is what happens in real COVID-19 disease. And that's how this vaccine is protective against COVID-19. Thank you so much for watching this video.